and look at the bottom two. You have a charge there, plus, right? Some have plus, meaning they are charged. Those atoms are charged. So whenever you say polar, that means there is some charge on the component atoms. So polar amino acids carry a charge, non-polar they do not carry a charge. Again, I could ask you to memorize the structure, but I'm not going to do that. But eventually you'll have to at some point. Maybe when you take chemistry. Okay.
On the top picture, there's no charge there. How is that a polar amino acid?
Cysteine, I don't know if they missed or not. I'll have to look that up. Normally, the charged amino acids should have a charge or some residual charge like in water, which is not a typical plus or minus, but they do have some unexplained charges, some kind of attraction. And the way the electrons move around when they are near each other, whether they are closer to the nucleus or far away from the nucleus. So when you bring two amino acids together, okay, what happens? You have a bond formation. In this case, what kind of bond? Can you tell from what you see here in the middle? You are bringing amino acid 1 and 2 together and you are forming covalent, because you see the line between the two, the single line. So the dipeptide forms. You have two amino acids joined together, and the peptide bond is made between the the OH of the previous one and the H. They are removed, the OH and H, because we are making something, right? Remember the dehydration reaction, the synthetic reactions have to remove water. So the OH from the first and the H from the second one are removed, so water is removed, and the bond forms between the CO and the NH. Can you see that? I want to give you a minute or two to understand, because these are all basic things, but these are going to help you further down. But only thing it's beyond the scope to get into how that happens and all that becomes chemistry, and that's beyond my time and scope. So that's where you need to refresh, go to some YouTube videos and look up the bond formation and things like that. Then you have to go to the table, look up the number, you have to draw the sketch, and so that's how you understand how this happens. But for our purpose, the bond occurs by removing water because it's an anabolic synthetic reaction, and you are forming the bond between the CO and the NH. They call it carboxyl and the amino group. So every amino acid has a two end. It has an amino terminal end, the carbons, and the carboxy terminal end. They call it amino and carboxy or carboxyl terminal. So if you have another amino acid like that, so the bond occurs between these two. That's what they have explained there. Okay, we'll be removing one of the hydrogens from this NH3, and the water will be removed, and between the CO and the leftover NH, you have the bond formation. The bond is going to be called peptide bond, because you have two amino acids. If you have only one amino acid, you are going to call it amino acid, period. If you have two, it's going to be called dipeptide. You are going to have three, what do we call that? Tri, tetra, and so on. If you have several, up to 10 to 20, you can call oligo, like oligosaccharides. If you have more than that, polypeptides, like polysaccharides, monodisaccharide. We talked about that the same way. So So in the book it says 10 to 15. Okay, so that will vary. So if it's around 10, just for consistency, because for the sugars they say 10 to 20, here it's in the book it says 10 to 15. So we can keep, if you have a number around this, if you have much higher, very high numbers, like, then you can consider them polypeptides. Protein structure conformation. So the protein structure is very vital. Okay, it's like you have a blueprint. You start building your NS building or your new bachelor's program building. So you start putting the blocks together, and finally you have something that you can go and sit in the classrooms, right? So it involves a lot of work. So your proteins go through several steps. So they call them primary structure, secondary structure, tertiary structure, the third level, and the final 3D structure is the quaternary structure. Can you guys see where you find these words? If you need to look up the figure, turn over the page, 68, you can see the different levels of protein structure formation. Okay, so what do we see? The first one, a sequence of amino acids like beads on a chain, the colored beads. That's your first structure. Okay, like you put the amino acid beads together. Okay, then you can fold them like this, or you can make different structures, as you can see. One they call helix, the coiled structure. They even call it alpha helix, the way it is twisted. And these ones they call beta sheets, or beta pleated, meaning folded sheets. So this is the second level. You take this long chain and you make it spiral, or you make it in a different form. So they take either form, as you can see. One is the alpha helix and the other one is the beta sheet. The third level, what is happening? You have a combination of the two, random combination. So that is your tertiary level. And then the final quaternary level is the final protein structure. So here they have the hemoglobin, and you have two in golden color, two in purple color, right? Each one is a peptide by itself. So hemoglobin has 4 polypeptides, and the red star-like structure, that's an iron molecule. So each one has an iron in the middle, and this is hemoglobin. Hem means iron. Okay, so this is the final structure of hemoglobin, for example. So like that, every protein has a definite structure, and that structure can change even a little bit. Sometimes it doesn't affect the protein, sometimes it can lead to serious conditions, depending on the case. So on figure 226, for this try to know which one would be the primary, secondary, tertiary, quaternary structure if you are given those examples. And then they are given protein functions, how proteins function in our body. They are structural, they help to communicate, they help to transport proteins, as you saw the proteins on the gated channels on the membrane, right? And then they help with catalysis, facilitating reactions in the form of enzymes. They serve as catalysts. Earlier I mentioned an example for catalyst. Recognition and protection, movement, cell adhesion. So at least know the major functions, how they play a role. And the examples you can look up and read for yourself. You don't have to memorize, because some of them you may know, some of them you will know better as you finish this course, not now, because you haven't come across some of those terms. Keratin, for example, is in the hair, so, but know the functions. Figure 226 shows insulin made of 2 polypeptides. It's a very simple structure compared to hemoglobin. Then you have enzymes and metabolism. So I'm going to talk about that a little bit, but I will brush up on that in the next class, and then we will jump into chapter 3. So you need to be reading up on chapter 3 from now. Chapter 3 and 4 if somebody wants to be ahead of the curve. So enzymes, what are enzymes? They are catalysts. What are they made of? Enzymes are proteins made of amino acids. Okay. What else enzymes do? Enzymes facilitate chemical reactions. Sorry?
Are they catalysts?
Yeah. I said that catalysts are proteins made of amino acids. Enzymes facilitate or speed up reaction. So some reaction with the enzyme goes faster than without it. Some reactions, they don't even happen without the enzymes. Okay. And even the enzymes need some help. We will talk about that. Okay. So enzymes act on some substance and change it to something else. So you see the word substrate. Substrate is a substance that is being acted upon by the enzyme. So usually an enzyme gets the name based on what substrate it's acting on or what product it's forming. So you will see when you see some of the names of the enzyme and the action it does, you'll get names. And let's turn to figure 228. You have an enzyme that's binding to sucrose and breaking it down to the component units. So you see the enzyme at the bottom, the purple color, what's the name of the enzyme? What's the name of the enzyme? Sucrase. So most enzymes end with the three letters ASE. So in this case, sucrase. Why? I told you, the enzyme gets the name from the reactant or the product, or the substrate or the product is, in this case, is working on what? Sucrose. Be specific. Sucrose. So you are acting on sucrose, so the enzyme gets the name sucrase. Okay, the sucrase breaks down the bonds between the glucose and fructose that make the sugar. So when I say glucose and fructose, where did that come from? See, that's why we need to remember what we already talked about in class or what is discussed earlier in the chapter. Sucrose is made up of glucose and fructose. So you can see the glucose and fructose there, right? So the bond is broken, they are held in place, and they are held in a site where all the action is happening, so they call it active site. So the substrate or the substance is bound to the active site in an enzyme, and then this reaction occurs. So what's the reaction? This is a decomposition type reaction where you're breaking a bond and you're releasing the glucose and fructose. This can be reversed. Okay, so if the reverse happens, it is an anabolic type reaction. So an enzyme can bring glucose and fructose and tie them together. Okay. Some enzymes do only one way, some can do both ways, as the case may be. And then if you look the graph on 227, you see the red line. Imagine that's a hill, okay? And you see a section that's green in color, it says active energy, not active, activation energy. So think this hill is, okay? There is a hill like this and there is a hill like this. It's easier to climb on hill A or B?
B.
B, because the slope like your treadmill, okay? So you need a lot of energy if you have to go on this way compared to this way. So if you have the enzyme, it's like having the alternate option, okay? So the enzymes reduce the energy that you need, meaning for the reaction to proceed, okay? So the activation energy, the green area is much less on the right hand side compared to the left hand side, meaning enzymes facilitate and speed up reactions by reducing the energy for the reaction to proceed. What kind of energy? By definition, activation energy, meaning to activate the reaction. Okay.
So
Then,
would enzymes be considered under, like, would it fall under the category of a catalyst? Or
yeah, we
would-
said that 3 times. I told him, and now I'm.
I
No,
understand
no,
that it is.
yes, your answer is yes, and we said that 3 times. Now, all I'm saying is that you need to be more attentive.
I am being more attentive. I heard you
Okay,
say
I
it
saw
three-
you getting on the phone, I didn't say anything,
Okay.
okay?
And I'm
That's
looking up what
breaking
enzymes
the
are.
policy. Let's not ask for more trouble, let's just cut it out there. Okay, because you're a good student, I want to have you like that.
Right.
All right, period. I'm watching everybody what you do as you sit in the class. Then we talk about cofactors, metabolic pathways. Metabolic pathways tell you which way the reactions proceed and how the different components are being modified. And for all that, you need energy. Energy is in the form of ATP, adenosine triphosphate. If you don't know what is adenosine triphosphate, please look it up. Okay, that's the basic energy molecule for the cell in order to function. Okay, so we will talk a little bit about ATP and then move on to chapter 3 when you come back. Okay, thanks for your attention and have a good evening.